Hello and welcome to this teaching point about light and optics. Optical elements in a flow cytometer are important because they help us understand where the light is going to go, which helps us understand where our fluorochromes will be measured in our system and what fluorochromes we can use. So let's start talking about some principles. Refraction is the principle that light will change direction when it travels from one medium to the other. This is described by Snell's law. Refractive index is something you may be familiar with from microscopy. Is a number that describes how light propagates in a given medium. This is an important concept for us in flow cytometry because we have transitions between air and glass or quartz, and then quartz and water to measure our cells. Diffraction is a concept also from microscopy, bending light around small objects. If the diameter of our object is wider than the wavelength, the light will just pass through. However, if the diameter is smaller, we see this diffraction patterns as shown here. Diffraction is also the principle behind the Abbey diffraction limit, which describes the theoretical limits of resolution based upon the wavelength. Another property of light is the concept of scatter. Where light interacts with a particle, that light can be scattered. Rayleigh scattering is a scattering of light in a media without changing wavelength. This is useful for some new instruments coming out that are looking at cells using label-free methodologies. In flow cytometry, we focus on something called scatter, which is the scattering of the light by particles that have a diameter greater than or equal to the wavelength of light. The larger the particle, the greater the scattering. So this is why we say forward scatter. The larger particles have a higher forward scatter. Next, we should think about our light sources. And predominantly, our systems are using lasers or light amplification by stimulated emission of radiation. Laser light has several properties. It's high energy, which is good, polarized, and it's coherent, both spatially, which means there's little divergence of the beam, and spectrally, which means it's usually of a single wavelength. Here's an idealized laser. Energy pumps the laser in the gain medium. The molecules in this medium get excited, and they start emitting their own photons. Those excite other molecules, and that process continues until energy is of sufficient power to escape through this output coupler here, typically a half reflector mirror. Now that we've talked about properties in light sources, let's talk about filters. Filters are important because they help us control where light goes in our flow cytometer. We use things like short pass filters, which allows light below a stated wavelength to pass through. We use long pass filters, which allow light above a stated wavelength to pass through. We also use band pass filters. These allow light within a range to pass through. For example, here we have a 630-20 bandpass filter, which means centered at 630 nanometers with a 20 nanometer window, or in this case between 620 and 640 nanometers light, will get transmitted through this bandpass filter. The other thing that we use are dichroic mirrors. Typically, these are coupled to a long or short pass filter. They will transmit light based on the type of filter that's used. So for example, here's our 540 dichroic short pass. Light below 540 nanometers is transmitted. The light above 540 nanometers is reflected away. And this allows us to control light through our system. Now, if we take a look at an optical arrangement shown here and here, we have focusing lenses. These focusing lenses, shown here, help us shape the light and focus light in our flow cell, the optimal point where our lasers can interact with our cells. We also have collection lenses, shown here, and these collect the light and focus the light and transmit it to our collecting optic. Now, if we put all this together, the two important things that you need to understand are your excitation light sources, because this is going to help you understand what fluorochromes can be excited by your flow cytometer and potentially identify issues. Here's an example of such a case. Here we have PE and Percy-P Psi 5.5. The blue line here represents the Percy-P Psi 5.5 excitation. You see the maximal excitation is around 488 nanometers, but there's about a 40% efficiency at about 405 nanometers. So if you have these two lasers on your system, you're going to have to make sure and be careful when using Percy-P in your panel design. PE, likewise, has three excitation lines, 408, 488, 532, and 561. And if you have two of these lines on your system, you'll have to, again, be careful when you design your panels using PE. Now, the filters are important because they help you know where the fluorochromes are going to be detected on your system and hopefully identify potential overlap issues, which are going to require us to compensate. Here we have a fluorescence, the emission profile shown in dark green, with a 530-30 bandpass filter, which gets about 48% of that curve, and a 585-42, which gets about 12% of the curve. The fact that we have about 12% emission in this filter means that if we have a single fluorescein labeled cell, we're going to see signal in both of these detectors. So therefore, we're going to have to control and remove the fluorescence in this detector by compensation.